Welcome. This video will be about mesenchymal stem cells for immune diseases and anti-aging. The number of mesenchymal stem cells in our body decreases dramatically as we age, from 1 out of 10,000 cells when we were children to 1 out of 250,000 cells when we reach adulthood. This decline comprises our body's immune system, which leads to a higher likelihood of infections, cancers, and other immune or age-related diseases. Naturally, scientists have been working to find out how mesenchymal stem cells, MSCs for short, can be restored in the body to rejuvenate the immune system and prevent immune aging. This sounds easy, but the path to MSC-based rejuvenation is loaded with challenges. We are not just limited by scientific understanding, but also cell therapy economics and governmental regulations. Before we begin, I have to mention that MSCs have other abilities, such as being able to differentiate into other types of tissue cells. But today, we will only focus on the immunomodulatory aspects of MSCs. Now, what is a mesenchymal stem cell? It is a fibroblast-like cell that supports connective tissue. It can be sourced from many parts of the body and can differentiate into other types of tissue cells, depending on the specific culture type. We can find MSCs in bone marrow, umbilical cords, fat cells, and dental pulp. There are some variations in biological properties, depending on the source of MSCs, but they are all able to repair damaged tissues through a variety of mechanisms. MSCs are known for a homing ability, where it is able to migrate to the site of inflammation, and once it has arrived at its destination, it has mechanisms that include immunosuppression, anti-apoptotic, anti-inflammatory, anti-fibrotic, pro-angiogenic, anti-tumorigenic, neuroprotective, and antibacterial effects, all of which are positive. MSCs can be understood as a hit-and-run group of rescue cells. They show up at a disaster area in the body, control the situation, then leave after the area stabilizes. Now, aging has a large impact on our immune system. Aside from it being known as a natural process, aging is still not yet fully understood. 3 important hallmarks of aging are the shortening of our telomeres, increasing instability in our genes, and cellular senescence, where cells shut down and stop functioning, but damages other cells around it because they don't die off. These hallmarks come from a variety of factors, such as DNA damage, oxidative stress, prolonged exposure to inflammation, and many other factors. Through mechanisms that are still unclear to our scientists, an aging immune system, through the cellular senescence of immune cells, is called immunosenescence. Immunosenescence impairs the functioning of immune cells and causes various diseases as we age. We can categorize immune cells into the adaptive immune system and the innate immune system. The adaptive immune system is damaged a lot more from immunosenescence. This includes our T cells and B cells. B cells secrete antibodies, present antigens to other immune cells, and regulate T cells. Aging, in this case immunosenescence, reduces our B cell production and therefore reduces its effectiveness of our antibody response in the event of an infection. B cells are very important for vaccines because they remember viruses and create appropriate responses. But as shown in elderly people, B cell and germinal center responses are impaired after vaccination. T cells, likewise, decrease in numbers due to telomere shortening and impaired DNA repair. CD8 positive T cells that kill foreign cells experience a decreased ability to multiply, lower mitochondrial function, and increased reactive oxygen species that damages the cell with too many signaling molecules. On the other hand, innate Innate immune cells contain monocytes, macrophages, neutrophils, natural killer cells, and dendritic cells. Immunosenescence decreases our macrophages' ability to consume foreign cells and accumulates M2 anti-inflammatory macrophages and converts them into pro-inflammatory macrophages. Immunosenescence also decreases our neutrophil and NK cells' ability to fight pathogens. Dendritic cells are the least affected by aging, but it does cause an antigenic shift for dendritic cells in aging into an inflammatory state, causing immune system imbalance. In some, there is a death spiral where aging causes a decrease in strength of the immune cells, which increases inflammation, which leads to further aging of the body. Can mesenchymal stem cells reverse this trend? Clinical researchers have already proven its ability to combat autoimmune diseases, including Crohn's disease, lupus, and graft versus host disease. But how exactly do MSCs perform their immunomodulatory functions? We know that after being airdropped onto the site of injury, MSCs will conduct its operations in two main ways. Number one, intracellular liaison, or also known as cell-to-cell contact, and number two, paracrine signaling, which refers to the secretion of bioactive molecules. You can think of them as a temporary operating base at the site, but instead of one operating base, there are millions of MSCs working at the same time as operating bases. Let's start with cell-to-cell contact, intracellular liaison. In immunology, we would categorize in two ways, just like our immune system, adaptive and innate. When faced with inflammation, MSCs upon physical contact with immune cells, like naive and memory T cells, were able to suppress their reactions against antigen-presenting cells, APCs, that serve to activate them by communicating the presence of infection. MSCs do this with gluing T cells on them using VCAM1 and ICAM1 proteins, neutralizing them and halting the inflammatory response. In another case, they may trigger Notch1 or FOXP3 pathways in T cells, turning them into regulatory T cells that reduces inflammation, or produce a protein called galectin-1 to suppress T cells. MSCs also prevent B cells from apoptosis, cell death, by freezing them in place. B cells in contact with MSCs remain alive, but stop dividing or producing antibodies. They are, in effect, caught in a state of suspended animation. The MSC does this by inhibiting their PERK and P38 growth pathways. On the other hand, if there is a need to provoke a stronger immune response instead of suppressing it, MSCs promote Th177 T cells to fight specific pathogens. MSC cells have toll-like receptor proteins TLR3 and TLR4 on their surfaces that can detect viruses and bacteria, switching off its immunosuppression ability and instead boosting inflammation to fight infection through the secretion of cytokines. Scientists have been able to use MSCs to help the body's T cells fight cancer by boosting adaptive immune cells, or conversely, suppress inflammatory responses like during pregnancy loss through the introduction of IFN gamma, a pro-inflammatory cytokine found in T and NK cells. MSC's influence on the innate immune cells are less researched, and this makes sense because MSC's influence is less impactful in this area. It is known that most unengineered MSCs are first consumed by immune cells in the lungs after being intravenously injected into the body. MSCs are also able to activate pro-inflammatory M1 macrophages into anti-inflammatory M2 macrophages through a lipid signaling molecule called PGE2. They are also able to feed neutrophils ICAM1, deactivating them and preventing tissue damage. Aside from cell-to-cell contact, MSCs can also communicate messages by releasing factors in its secretome. The secretome contains cytokines, growth factors, chemokines, extracellular vesicles containing microRNAs, for example, but we won't get into the technicalities of all these molecules. What is important is that we understand that all these factors come together to regulate the immune system, communicate with other immune cells, and support tissue repair. For the adaptive immune system, MSCs are able to release interleukin 10 and PGE2 that shuts down Th17 cells, T cells, alleviating inflammation. These molecules alter the epigenetics of the T cell through histone trimethylation to stop them from producing inflammatory cytokines like TNF alpha, interleukin 17, 22, IFN gamma, and others. MSCs also use the well-known PDL1 protein ligand to deactivate T cells. The MSC will use IDO, an enzyme that suppresses inflammatory T cell activity and encourages the growth of regulatory T cells, Tregs. For the innate immune system, MSCs uses IDO enzyme and PGE2 to suppress NK cells. If required, MSCs can boost NK cells' ability to produce IFN gamma. MSCs have an interesting effect on neutrophils, our body's first responder white blood cells. MSCs release a soup of molecules to boost the fighting ability of neutrophils. These instructions come in the form of exosomes, molecules wrapped in cell membranes, or cytokines like MIF, interleukin 6, interleukin 8. Because getting MSCs to be consistent is extremely difficult, scientists have toyed around with preconditioning MSCs in order to produce a specific outcome of MSC when delivered into the body. This includes priming or licensing MSCs to make them more potent through hypoxia, growth factors, heat shock protein, liposaccharides, pharmacological agents, chemical agents, serum deprivation, or inflammatory stimulation with cytokines. Many of these treatments are proven to be effective against disease models as they mimic the disease environment. We can also combine the preconditioning, such as treating MSCs with hypoxia and inflammatory cytokines, into new ways of MSCs. So I expect them to be able to create super MSCs very soon that are tailor-made for any situation, only if research and VC funding permits us to experiment with recipes more often. Because the natural abilities of MSCs are often not potent enough, scientists have begun genetically engineering them to become targeted delivery vehicles for specific therapeutic agents. This involves modifying the MSC's own DNA to turn them into living factories to mass produce and release high concentrations of desired proteins, such as powerful immune signals or even oncolytic viruses designed to burst cancer cells. We now have MSCs engineered to act as cancer-fighting agents by locally activating killer T cells as anti-inflammatory surgeons for conditions like osteoarthritis or as regenerative engineers for tissue repair. We are also experimenting creatively with cryopreservation and using gene delivery vehicles to improve the ability of MSCs. But despite all these experiments and despite all the evidence for MSC's role in reversing the age of the immune system, we still don't have effective treatments, but countless biotechnology companies are working exactly on the problems mentioned, aiming to strike gold with the right drug and further encourage research and investment into the field. However, the administration of MSCs is still a novel therapeutic strategy. It will take some time before we achieve results in both clinical and preclinical and clinical research. The more we learn about MSCs, the more we don't understand how its mechanisms work to produce different immunomodulatory effects. Because we don't understand how MSCs work, it is still difficult to explain the differences in clinical results. For example, clinical trials can produce varied immunomodulatory effects depending on in vivo or in vitro experiments or whether MSCs were allogenic or autologous, and a number of other factors. I like to think of MSC research in drug discovery as similar to the early 20th century, where scientists with little to no understanding of how chemical compounds worked still were able to create drugs purely through trial and error. That was how drugs like penicillin and aspirin were created. And it would surprise many that when those drugs were invented and commercialized, we knew very little of how they worked, only that they were effective. I would say that it is only a matter of time that MSC preconditioning, genetic engineering methods, and combination with other drugs are optimized to the point where we can enjoy off-the-shelf, affordable MSC treatments that reverses the damage in our aged immune systems. Thank you for listening. If you are interested to read more, the links to the reference material are in the description. And please let me know in the comments if there are any issues.